So I'm going to be talking about cancer evolution today from an organismal perspective. And the work that I'm presenting is part of a sort of a very integrated group that I work in that works on epidemiology, and I handle the comparative genomics. And we also have functional validation of the targets that we find through this research in the lab. What I'm going to focus on today, though, is the comparative genomics and some projects that my students have worked on recently, because I'm really proud of them. So I'm going to walk us through a background of cancer evolution from this organismal perspective in terms of life histories, and I'll describe Peto's paradox. And then I'll postulate some potential mechanisms of cancer resistance that we see across the tree of life. I'll then talk about our recent research into cancer phenotypes across vertebrates. And then I'll talk about a study where we looked at gene copy number and malignancy rate. So one thing that we see right across the tree of life is that we see cancer phenotypes, right? So from plants to fungus to mammals and invertebrates, okay? What this suggests is that cancer has probably been a selective pressure on organisms since at least the origins of multicellularity. So it's quite an ancient phenomenon. And so as evolutionary biologists, what we're really interested in, at least from this organismal perspective, is that if this has been such a pervasive selection pressure for so long, why hasn't natural selection eradicated cancer? Why do organisms still get it? And so this is a sort of a guiding question that promotes all this research for us. And we think it's helpful to think of this from a life history perspective. And that's because resources, of course, are limited in nature, and organisms face trade-offs over where to devote those resources. So the reason, right, is so that they can have so they can increase their fitness that allows them to survive at least a reproductive age. So theoretically, an organism, trying to find my, yes, okay. Theoretically, an organism can devote all of its resources to somatic maintenance, right? And its important functions like DNA damage, repair, cell cycle control, and immune function. And that would probably overall lower its cancer susceptibility. But that would take away from really important investments in reproduction, which of course is extremely important for maintaining organismal and evolutionary fitness. So the type of cancer defenses that an organism might be adapted towards really depends on where it exists on this spectrum. So we like to think of it in terms of fast life histories versus slow life histories, right? So you can imagine a mouse, right, is going to be devoting a lot of resources to producing many offspring before it itself gets gobbled up by some predator somewhere. And it's not really worried about living a long life and having to deal with cancer eventually. Meanwhile, on the other end of the spectrum, a humpback whale or an animal like it is going to live a much longer life and have much fewer offspring, right? And it's going to have some kind of cancer defense mechanism so it can live long enough, right, and be a great grandparent, essentially, right? Another way to look at it is from the perspective offered from Peto's paradox. So this is coined after the epidemiologist Richard Peto, who demonstrated using mouse models that the probability of cancer happening in those models was really directly related to the length of time at which these models were exposed to carcinogens. So what that leads us to ponder about is that if animals with a large number of cells and extremely long lifespans, right, let's say 10 times greater than a mouse, right, why aren't humans, for instance, right, 10 times more likely to get cancer than a mouse? So this is the essential question of Peto's paradox. And evidence for this paradox lies in the fact that large animals like elephants certainly get less cancer than humans. And also, animals like the naked mole rat, which is a really unique organism. This is a mouse-sized animal that lives about 30 years, and it's well known for having even though a history of thousands of necropsies over the decades, really no instances of malignant cancers that are known in this species. So for us, right, we like to think of it this way, right? So there are algebraic models that can accurately and precisely predict colorectal cancer rates in mice and humans. And these models are based on like the number of crypts in the colon, the mutation rate per cell division, and so on. But what these models also predict, while they accurately predict these colorectal cancer rates for mouse and human, they predict that all whales are going to have colorectal cancer by the time they're 90 years old. And so we know that this probably isn't the case, right? So the question is, how do whales, right, flatten this curve, right? We're interested in this space here, okay? So solving Peto's paradox, right, from this perspective, it could potentially tell us how natural selection has beat cancer, at least long enough for an organism to live beyond reproductive age. So it allows us to attempt to answer this question, right? What are potential mechanisms of cancer resistance found across nature? So we sequenced the humpback whale genome a while back, and we compared it to the genomes of other cetaceans. So cetaceans are the mammalian order that include whales and dolphins. And what we found is that when we looked at positively selected genes that were unique to the baleen whale lineage, they included many genes that are known to be mutated in human cancers. So what this suggests is that as whales evolved their gigantic sizes, there was very strong selective pressure on these types of genes, possibly to help control DNA damage response and cell cycle control as they evolved those gigantic sizes. I'm going to talk about potential mechanisms of cancer resistance here, and the first one I'm going to introduce is this possibility that organisms might lower their somatic mutation rate. And what that would do is basically reduce the overall number of hits in a clonal lineage during the lifetime of an organism. So when we sequenced the whale genomes, we did find that over millions of years, whales have accumulated far fewer DNA substitutions in their genome than other mammals. And so this suggests perhaps lower germline mutation rates in whales. However, also when we were able to collect data from other studies that looked at somatic mutation rates across mammals, we found that somatic mutation rate was really strongly correlated with neoplasia prevalence. And I'll talk about neoplasia prevalence in a moment. But what this really means or suggests is that organisms with lower somatic mutation rates do get less cancer. I'm going to briefly throw this up here because I know Stefan's in the audience and I want to show him this. My PhD student, Vahid Fard, has been collecting potentially active line and sign elements from a number of mammalian genomes for which we have necropsy records. And we found some interesting things. One thing is that the number of potentially active line 1 elements in the genome can predict neoplasia prevalence in the genome. Right? So this suggests, right, whereas I was talking about nucleotide substitutions in the previous slide, other types of structural kind of mutations like transposable elements might be having an effect here. But what's also interesting is when you add signs, right, now signs depend on lines for their movement around the genome. When you add signs into the equation, you can predict malignancy prevalence. So what this suggests is that while lines can help you predict neoplasia, it's the movement of the signs that depend on the lines that actually help you predict malignancy. Okay, so I'm going to move on to another potential mechanism of cancer resistance, which would be a more sensitive or efficient apoptotic response. So in this case, cells might be more likely to actually self-destruct in the face of DNA damage, thus stopping cancer dead in its tracks. Now, we know when we compare the apoptotic response to DNA damage between humans and elephants, we know that elephant cells are far more likely to undergo apoptosis when they're exposed to this DNA damage. So what this suggests is the potential mechanism of cancer resistance in elephants is the fact that their cells are just far more likely to blow themselves up and stop cancer. So we think that's pretty cool. I'm going to now talk about a third potential mechanism, which is a redundancy in tumor suppressor genes. And it's somewhat related to the apoptosis I mentioned on the previous slide. What this would do is require more mutational steps for cancer to initiate, which would allow the organism to more effectively live out its lifespan. So TP53, right, is the best known tumor suppressor gene. It's mutated in the majority of human cancers. And TP53 is called the guardian of the genome, and it's really essential for triggering cell death or apoptosis in the face of DNA damage. So human individuals with Li-Fraumeni syndrome have a mutated allele of TP53. So they only have one functional allele. And individuals with this condition face over a 90% lifetime risk of cancer. Also, cells from Li-Fraumeni patients, or Li-Fraumeni cells, right, are far less likely to undergo apoptosis when they're exposed to DNA damage, especially compared to healthy human controls, which have two functional TP53 alleles. Now, we found that the elephant genome has over 20 copies of the TP53 gene in their genomes, so potentially 40 alleles, right? And elephant cells, as I mentioned earlier, are far more likely to undergo apoptosis. We found that this high number of copies of P53 is shared between all elephants that we've studied so far. That includes extant elephants like African, Asian, and forest elephants, but also the straight-tusked elephant and the woolly mammoth, which are, of course, extinct. So what this suggests is that this duplication of TP53 occurred in a common ancestor of these elephants. And when we did a phylogenetic analysis of P53, not only its the functional homologs, but also all of the pseudogenes and other copies that we found across mammals, we realized that a duplication of TP53 occurred in the ancestor of manatees, hyraxes, and elephants. These are called penungulates. And so we think that P53 duplicated in the penungulate ancestor, followed by a huge expansion that's unique to the elephant lineage. So what that means is that as elephants got larger, they added P53 to their genome. Okay, so redundancy in tumor suppressor genes like TP53, it could be a common form of cancer suppression, but we don't really know. So the next question we asked was, does this tumor suppressor gene copy number, does this play a role in cancer resistance across mammals, right? Not just elephants, but what about the diversity of mammals that we know of? So what we wanted to know is could we find an association between gene copy number and life history traits that we know are associated with cancer? These life history traits are lifespan, body size, basal metabolic rate, other things that we collected from databases. And for gene copy number, in this study at least, we focused on tumor suppressor genes and oncogenes that are known to be mutated in human cancers based on the Cosmic or Cancer Gene Census database. So we're able to collect a large number of mammalian genomes from NCBI, and essentially we formulated a sort of reciprocal blast search that uses the human protein to search for copies in the mammalian genome and then collect the putative paralogs from that genome and then blast them back to the human protein database to validate their orthology. And then once we collect these copy numbers, we use them in a PGLS analysis, or phylogenetic generalized least squares analysis, which can help us determine the association between the copy number and the life history traits while taking into account the phylogenetic relatedness. Okay, so in this study, right, we were able to collect 63 mammalian genomes that are representative of mammalian diversity in terms of phylogenetic diversity, but also in terms of the sort of breadth of body mass and lifespan that we see across mammals. And we were able to take the tumor suppressor genes and the oncogenes and count their copy numbers in each of these genomes. Okay, so the winners, right, are the naked mole rat, right? So these are the three species that were the top in terms of the number of cancer genes in their genome. The naked mole rat, I already mentioned this animal, so we were very excited about this result. It's very low cancer animal, and it also has the most tumor suppressor genes in its genome across the mammals that we looked at. Not much is known about cancer in armadillos and sloths, but we think these animals, and I'll show you later, also might make really promising models in cancer research. Okay, one interesting finding was that the number of tumor suppressor genes in a genome is really highly correlated with the number of oncogenes in the genome. And this was an interesting result for us. What we think this might mean is that it might reflect the fact that there are these complex regulatory networks that really mediate that balance between cellular proliferative potential and checks on tumor progression, which is sort of suggested of those trade-offs that I mentioned earlier. So this is happening at the genomic level as well. Interestingly, we found that it's longevity that predicts tumor suppressor gene copy number in a mammalian genome rather than just body mass. So for instance, right, these very small mammals, right, naked mole rat and these Myotis bats, they're small, but they live a really long time, especially compared to other mammals that are the same size as them. And they also have many tumor suppressor genes in their genome. So what this suggests is that every time extreme longevity has evolved across the history of mammals, it's been accompanied by strong selection against these, for these extra safeguards, right, against both hereditary and sporadic cancers in these long-lived species. And we're seeing these patterns in the gene duplication. Now, to look at this, right, we use this term longevity, but what we're looking at is called the longevity quotient, which is essentially the difference between an animal's observed longevity and its expected longevity, right, given how big it is, right, compared to other animals of the same size. So I just want to explain this for a moment. So humans are here, right? You can see we're a little bit above, in terms of our longevity, where you expect we might be given our body mass. That's perhaps not surprising because we have modern medicine, right? But the elephant lays right on this line here. So the elephants have a longevity quotient of 1.0 essentially, which basically means they live exactly as long as you would expect given how big they are. These other species up here, however, right, are generally small mammals, right, that have very high longevity quotients. So these results are showing us that these species, or species like this, might be prime targets for cancer resistance research. Okay, so we've shown an association between cancer-related life history traits and cancer gene duplications, but what about the cancer phenotypes themselves, right? How does cancer prevalence vary across vertebrates? Now, this is not an easy question to answer. There's not much information about cancer across species. Most cancer studies come from model organisms, and there hasn't really been a systematic look at how cancer pops up in different organisms across the tree of life. So we've been working on this for a while. I've been part of the Cancer Evolution Center, I think now for 9 years, with Carl O'Malley and Amy Boddy. And we've been working to collect pathology information from accredited zoos across the world. And we now have health records for over 170,000 animals, from about 13,000 species. And this has been a very long-term project, but the grad student that eventually inherited the project and took it to its completion is Zach Compton, who's really great, and he's now a postdoc. Okay, so we published a major study last year. When we focus on just the species for which we have 20 or more necropsies in the database, we're able to collect 16,000 individual animals for 292 species, right? So it gets a lot smaller, but we get a lot more power there. When we focus on just these species, and these are mammals, reptiles, birds, and amphibians, we found that across all these animals, neoplasia prevalence was about 4.9%, while malignancy prevalence was about somewhere around 3.2%, so somewhat lower. But when we look at and compare neoplasia and malignancy prevalence between the major groups of vertebrates, so amphibians, birds, reptiles, and mammals, we see some different patterns here. And what I just want to highlight, right, is that mammals overall have the highest prevalence of both neoplasia and malignancy. And so our own species' battle with this disease might actually be an artifact of our evolutionary history as mammals. So here I am, right, showing you the diversity of neoplasia and malignancy prevalence across the mammals that we looked at. And that's indicated by the green and purple bars here. And so you can see those prevalences vary widely across the mammalian phylogeny. But what I want to highlight, right, which I think is really cool, is that some of the species with the lowest cancer prevalences, right, sometimes 0 neoplasias detected, are actually the exact types of species that we expected given the genomics that I just told you about earlier, right? So those include cetaceans, bats, and the sloths and armadillos. So that was really exciting to us. So what it means is that the genomics that we did can actually predict or help predict cancer resistance across species quite well. Okay, so whereas before we were looking at the relationship between gene copy number and life history traits across the mammals, okay, we're now able to actually look at the cancer prevalence, right? So we can use these cancer phenotypes now to see if we can predict, if gene copy number can help predict cancer prevalence. So in this study, whereas before we focused on tumor suppressor genes and oncogenes, it was a very human kind of oriented analysis, we wanted to expand to the whole proteome of the organism. And so for this analysis, we looked at all protein coding genes and not just tumor suppressor genes and oncogenes. And we collected them from the mammalian genomes, and we used a handy tool called OrthoFinder, which basically uses an all-to-all blast from the complete proteomes of multiple species and clusters them into these groups of orthologous groups that are called orthogroups. And this was headed by Sophie Matthews, who was a terrific PhD student from the University of Galway in Ireland, who came to my lab for 3 months to do this project, and less than a year later, we published the papers. Like, she's just great. And Vahid helped her also, so really proud of these guys. Okay, so what we're able to do is collect about 100 mammalian genomes, which overlapped with the necropsy database, okay? This sample, that we took is representative of mammalian diversity and also body size and lifespan. But it's also representative of the breadth of the different cancer phenotypes that we have, right? So that's neoplasia prevalence, malignancy prevalence, and then the ratio between the two of them, which we call the malignancy rate. And we found that when we did our PGLS, there were 15 orthogroups that were associated with the rate of malignancy. I'm not going to get into all of them because we'll be here like all day, so I don't want to do that, but I just want to highlight 3. The first one is this nuclear receptor coactivator 4, which is involved in the process of ferroptosis, which is really kind of cool because we know that cancer cells are very high in iron content. There's also TUSC3, which is a candidate tumor suppressor gene, but it also accelerates cancer growth, so that was pretty cool. And then this NEC3, which plays a role in cell cycle progression and cancer progression. Okay, so we expanded our PGLS analysis from studying the relationship between individual genes and the phenotypes to studying gene sets. And the reason we did that is because when you look at individual genes and their copy numbers, there actually isn't much variation across the sample of different species. And so that makes it difficult sometimes to detect an association, particularly when you're looking at so many genes, right? So in this case, we're looking at 20,000 mammalian orthologs. So there just isn't a lot of statistical power when you do it that way. So what we did in this case is what we call a gene set analysis, where we looked at the sort of aggregate count of genes that belong to different sets. And those sets are defined by pathways or processes. And so then we looked at whether these gene sets had expanded or contracted in association with body size, lifespan, and the cancer prevalence. And so when we did this PGLS analysis, okay, we found 10 gene sets that were associated with the rate of malignancy. What's interesting is most of these have a negative association with the rate of malignancy, okay? Except for the 2 with the asterisks here. Also, a few of these are associated with immunity, which is interesting to us because that's related to cancer as well. But this is the gene set that in our PGLS analysis has the strongest association with malignancy rate, so I'm going to focus on this too. And you can see it semantically kind of clusters with the other top ones as well. And that's negative regulation of transforming growth factor beta production, or TGF-beta. Okay, TGF-beta is a cytokine, so it's a protein that's involved in immune function and cell survival. It plays an important role in tumor genesis. Mainly at the onset of malignancy, it acts as a tumor suppressor. And it's identified as a potential target for cancer therapy. And so we found that increased gene count in this pathway is associated with low malignancy. And we use ancestral state reconstruction to look at the rate of evolution in the count of this gene set and the rate of malignancy. And I'm not going to get into it too much, but you can just sort of match the colors and you can see how these patterns kind of change in tandem over the course of mammalian history. I'm going to quickly run through this gene set analysis that we did. This is just another set of analyses in addition to the PGLS that I talked about. And this is a gene set enrichment and an overrepresentation analysis. And so what we did was we relied on the original tests I told you, like all those tests from all those different genes and the life history traits. And we were able to rank those whoops, where am I going here? Yes, we ranked those genes, right, according to their association. And then we also used simulations to set a p-value cutoff. And these are the 3 gene sets that were significant and in common with all 3 of the analyses I just talked about. The first one I already talked about, but then there's the TAP complex binding, and then also MHC class 1 peptide loading complex. So expansion of these are related to reduced malignancy across mammals. Okay, so I'm going to wrap up here, right? So to conclude, cancer is the original problem of multicellularity, and all species are endowed with some form of cancer resistance so that individuals can survive to reproductive age. And the discovery of species with the lowest cancer rates can elucidate mechanisms of cancer resistance. Our results show that in mammals, extreme longevity is associated with an overabundance of tumor suppressor genes, and also that gene copy numbers associated with the rate of malignancy, which pinpoints the pathways and processes that are involved in cancer resistance. Okay, with that, I'll end my talk. Thank you for listening. I want to highlight the work of these students and also my collaborators, and I want to thank the organizers of the conference. And I'll be happy to take any questions now or at a break. Thank you.